In this video, we will discuss about another paper which shows that most of the PDAC samples or patients that are affected with the pancreatic cancer cells has a high expression of MCL-1, SRC, and CFL-1, and this could be the reason they have a low survival rate. So the approach that they used in this paper is that they trying to suppress this MCL-1 expressions or the other um, genes expressions to see which of them will reduce the pancreatic cancer cells invasions. So first, in this figure 1, what they did is they validate the high expression of MCL-1, SRC-1 and CFL-1 from the online available data about the pancreatic cancer cells. So in the panel A, they shows the expression levels of these genes markers, that is SRC, CFL1, MCL1, in different database. First one they checked in the TCGA database, that is the Cancer Genome Atlas database, and they are able to find the mRNA expression of these genes are at higher level, and it is comparable to this Genome Cancer TCGA Pan database. So they checked it in the breast cancer and collagen CA TCGA pan cancer database, and they are able to see the expression levels are high for all these three genes. And when they check it in the pancreatic cancer cells TCGA database, they are able to see these expression levels are also higher in this case. And thereafter they check the survival rate with the high expressions of this mRNAs from the publicly available data, and they checked it for the lowest quantile of this MCL1, CFL1, SRC, and they are able to see that with the high expressions of the MCL1, they are able to see low survival rate, that is fourth quantile. They are able to see the low survival rate for the MCL1, and for this CFL1, the survival rates are lower, but they are not that significant as we have seen in the MCL1. And even for the case of SRCC, we are able to see low survival rates. Here we see the low survival rates in the third quantile compared to the first quantile. Thereafter, they check the relationship between this MCL1, CFL1, SRCC, MCL, and CFL1, SRCC, and what they are able to find out is that this SRCC and MCL1 has SRCC and this CFL1 has some kind of positive relationship, as its R square value is higher compared to the other case, but it is not close to 1. And this indicates that though this does not give us a good idea, but it shows that there is some kind of relationship between SRCC and CFL1. And after that, they check the percent survival data from the clinical database of the pancreatic cancer patients, and they are able to see that at a high MCL they are able to see low survival rate, and similarly for the SRCC high they are able to see low survival rates, and the CFL high also gives the low survival rate. But in the combinations, when they are able to see that when the CFL and MCL both are high, the survival rate are drastically slows down compared to the all three cases. And when they did the Western blot in many of these patients, what they are able to see is that BCL, XL, and MCL1 are present in most of these samples, and this could be and these patient samples are also correlate to the high invasion behaviors. So this from this they made a point that it could be this MCL1 and BCL XL's expressions, high expressions, might be causing them to acquire these invasive properties. At the same time, they also see the SRC activations in those kind of samples. That is, SRC5Y146 is shown in most of the samples, so indicating that these SRC pathways are also get activated, which also plays a role towards the invasions that we have discussed in that we have discussed, or we have touched upon that in some of our previous videos. So all this data in the data presented in this figure indicates that PDAC has a high expressions of this MCL-1, SRC, CFL-1, and all of them are somehow correlated to the high invasions properties. So targeting these genes could be a good idea to suppress those invasive properties of the pancreatic cancer cells. So we will discuss further results in our subsequent videos that are presented in this paper. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.